Okay, so now going back to modules, you can find the PowerPoint that I'm going through here in topic 4, FOXP2 lecture. I'm starting right here. And it's in 2 parts, but they are both going to be done today. So part 1 is about transcription and translation, and part 2 has more to do with genetic recombination and reminding you about how plasmids can be cut using restriction enzymes and things like that. So this is a really good tie everything together type study. All right, so our learning objectives for part 1 are to know the 3 types, or know how the 3 types of RNA function, be able to explain the following terms: promoter, RNA polymerase, triplet codon, describe the process of transcription, and describe what would happen if one factor involved in the process were missing, explain how all cells have the same DNA but don't make the same proteins, describe the process of translation and predict what would happen if one factor involved in the process were missing, and be able to predict the protein sequence if the corresponding DNA sequence is provided. So typically, language follows this pathway of development, but about 5% of people don't develop their language normally. This can be caused by genetic factors that can affect muscle control. It could affect parts of the brain that have to do with producing words. It could be that they have auditory problems, so they can't hear the words correctly, and they end up saying the sounds that they're hearing, which don't match what they're trying to say. But while this is our general guideline, some people who have hearing problems, or not hearing problems, but speech problems, end up outgrowing it. It's really common that if a child grows up in a household that speaks multiple languages, their speech is going to be delayed, but then when they do speak, eventually it is better, but it just takes some extra time to sort out which words go into which language, where if a kid only learns one language, they don't have that delay. It's mimic every sound you hear. So in this article, or this case study, comes from an article where back in 1990, they started studying a family they've coded as KE in London, where half of the family members have speech disorders. So we haven't really gotten into pedigrees yet, but this pedigree is basically a family tree, and any shape that is shaded in shows that they are affected by whatever disorder is being studied. And squares are males and circles are females. The way I remember it is that girls are curvy. So here you have a mother and father on the top generation, and then they had their offsprings, male, male, female. All of them got married, reproduced. So this son had a daughter and a son. This son got married and only had one son. This daughter got married and had 2 sons and a daughter. This is just made up. It has nothing to actually do with the family, but just so you could see how they would study it. And in this case, we see that every child who was affected had at least one parent who was affected. And this tells us that this is going to be a dominant type of mutation. Again, this will be next week. We talk about dominance and recessives. But when we see recessives, it would be like if it skipped this generation and then showed up in the child. That would mean that these people were carriers, even though they weren't affected themselves, and they could pass it on. Since this time everybody shows it, it would be a dominant disorder. So, according to the KE family speech test results below, on which test did the unaffected group do better than the affected group? So if we look down here, we see that the affected group is the grayed out columns, and the unaffected group are the white ones. So the categories listed on the right side: words, non-words, and oral movement. Words, you could compare them. Non-words, you could compare them. Oral movements, you could compare them. All of the little dots here are where the people actually scored. The bar is the average. So here, the average was here, even though somebody scored really high, somebody else had to score really low. And so you have, on these categories, where did the unaffected group do better than the affected group? And it should be all of the above, C, 1, 2, and 3. Because we see here, the white column is taller on words, it's taller on non-words, and it's taller on oral movements. So that shows that the people in this family had problems with multiple categories. It wasn't just one thing, like it wasn't a hearing issue. They couldn't form the words because their mouths weren't moving properly. There was a lot of things interacting here. So they went in and did neuroimaging of the members of the family. And on the unaffected people, you see on the left hemisphere of the brain, there's an area called Broca's area. There's also Wernicke's area. It's another language center on that same side. And it is lighting up because it shows that that's where the brain activity is. That is normal. And then the affected people had this kind of pattern where you didn't have it all in one part of the brain. They were having light ups all over their brain. So the Broca's area was not working properly in the affected members of the family. So by analyzing the KE family DNA sequences, the scientists found that the speech problem was caused by a mutation in the FOXP2 gene located on chromosome 7, region 7q31. So whenever we see FOXP2 in italics, that is the gene name. If it's not in italics, it is the protein. So later on, when they start talking about the mouse, they will have the OX lowercase, but on humans, it's all capital. So the FOXP2 gene makes FOXP2 protein. So they found that on normal unaffected individuals, if you sequenced it and we have A, G, A, T, blah, 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 it goes C, G, T at this location. That codes for arginine. The amino acid short symbol is an R. Then they have the affected people. Everything's the same except for right here. They have half of the people have J, half of the time they have an A, or G, not J, G and A. So that means they're carrying one normal copy of it and one mutated copy. If you swap that, the G to an A, instead of getting arginine, you now get a histidine in that place. And if we went back and looked at the side chains, arginine and histidine are quite different. So it's kind of was a big enough problem that it affected the protein structure greatly. So now that they found it, I'm going to go back and give you some reminder stuff here. DNA has a promoter region, and when they show it like this, these are exons, exon, 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 exons. The areas here are introns. They get cut out, they're in the way. You would have it coded into an mRNA. The ATG site is further in. Remember, you transcribe a 5' untranslated region because the ribosome has to get into place and everything before you start coding for your AUG start sites. Same thing at the other end. TGA becomes UGA, which is a stop site, and then you have a 3' untranslated region. That's where you're going to attach your poly A tail and stuff like that. So then you go from mRNA into the fully functional folded protein. Reminding you again about the differences between DNA and RNA. DNA is double-stranded, RNA is single-stranded. Both of them use C, G, and A, but DNA has thionine, RNA has uracil, and the sugar that is used for RNA is ribose, and it loses an oxygen to form deoxyribonucleic acids over here. So RNA is produced via transcription. So the RNA polymerase binds to the promoter. It then unwinds the double-stranded DNA to begin assembling the RNA nucleotides. So C's and G's bind together, A and T normally bind together, but because there's no T in RNA, it uses a U instead. And then at the end, it'll release the RNA transcript. So just showing again, the RNA polymerase is going along, matching with the template strand. The coding strand on top would look almost identical apart from that T and U swap. So the direction of transcription is always 5' to 3', but that's the new strand being made is 5' to 3'. The template strand has to go the opposite direction, 3' to 5'. So make sure to please pay attention to that. So first part of this activity says the KE family moms are very concerned about their kids. As soon as the babies were born, their moms took them to a DNA sequencing center to do a test. The next slide shows the FOXP2 DNA sequences of 4 newborn KE family babies. The bottom strand is the template strand. Mutated nucleotides are noted with red. Get a piece of paper and write down what the FOXP2 transcription product is on each one of the 4 babies. The bottom strand is to be transcribed. So it tells us based up here at the top, the promoter is in orange, the coding strand part is in brown, and then another yellow is for the neighboring gene. So on an unaffected individual, you have your normal promoter, then you have your transcription start site. This is where your mRNA would be getting made. So the mRNA coming from this one would be UAUGAUUA. And that would, if you notice, you could read the top strand and swap out those T's for U's, and that's what the mRNA would look like. So John here has a normal promoter and a normal coding region, but he has a mutation over here in a neighboring gene. Athena has a deletion where she has lost her entire promoter, but her coding region's all right and the neighboring gene's all right. Phil has a normal promoter, but he has mutations for multiple nucleotides inside his coding region. Neighboring gene is fine. Kathy has mutations in her promoter region, but the coding region's fine, and the neighboring region is fine. So in a more simplified version, John had a mutation here. Athena's mutation was that the whole promoter was missing. Phil had mutations in the middle of this. Kathy had mutations here. Which baby is less likely to have severe speech disorder problems? You have anybody with an idea? Or you're going to sit in silence and I will tell you. So baby John, here in A, is less likely to have severe speech disorder problems because he has a normal promoter. Yes, A. Thank you, Samia. If his promoter region is normal, his coding region is normal, he's going to get a normal FOXP2 protein out of this. He might have another mutation that's causing problems, but that's not going to affect his speech. Athena's gene is never going to get turned on because she doesn't have a promoter. Kathy's might get turned on, but at a different amount. So she might end up with more protein than she needs or less protein than she needs. Phil is going to get protein, but it's going to be different. We don't know how much of a change it's going to be between those nucleotides. So, for more review on RNA, on average, a growing mammalian cell consists of 5% mRNA, that's our messenger RNAs that code for proteins, 15% tRNAs, transfer RNAs that bring in the amino acids during protein synthesis, and 80% ribosomal RNA that make up the ribosome. So tRNAs and rRNAs never get translated into proteins. They stay and do their jobs as RNA. That's why I had you do the RNA folding handout, because you should see that where the strands can do binding to become double-stranded, that gives them a three-dimensional shape that lets them do work. So the tRNAs and the ribosomal RNAs are not proteins. They are going to stay as RNA. Only 5% of your RNA goes on to be translated into amino acids. So reminder, DNA goes through transcription in the nucleus to become messenger RNA, and then it gets transported to have translation in the cytoplasm because it has to attach to the ribosomes. We have our triplet codes. You should be familiar with this. We talked about it before. Remember the AUG, methionine, is our start site, and UAA, UAG, UGA are our stops, and everybody else just follows along. There's a translation video, but I do believe it's one that's already in your homework since it's from McGraw-Hill. It asks you to identify the players. So I'm going to give you a minute to look at this before I will tell you what the correct answers are. So you should be comfortable enough with the idea that down here at the bottom is your messenger RNA. This thing right here is a ribosome. Specifically, this is the large subunit, pointing on number 3. This is a tRNA on number 4, and it's holding the amino acids that are going to get.
Professor, quick question. You said be familiar with drawing this?
No, you don't have to draw it, but if I showed you a picture that you could be able to identify it, like, because the next part's going to have you like put them in order. And so you need to be able to go to identify it, that you go, okay, these are amino acids, that you have a growing polypeptide chain up here. So because sometimes they draw it a little bit different, and I want you to just be comfortable with this level. Like, this is the type of thing that will come out on like the test, that I'm not going to expect you to go into really detailed steps, but can you label APE? Could you label large subunit, small subunit? Where's the tRNA? Where's the amino acid? Where is the polypeptide strand? Where's the messenger RNA?
Okay, thanks.
This is just the level that you would be expected to understand. I don't want people, at the end of this, I am going to go through some review for exam 1, and just to help you understand the level that you should be comfortable for studying purposes. I don't want people learning all the different subunits from within the subunits and focusing on that kind of stuff. And it's like, no, this level, could you do this? So looking at the steps below for the growing peptide chain, could you put them in order here? And I'm going to tell you to focus on the colors and the number of of amino acids. So here there's 4 with 1 coming in. Over here there's only 4. Here there's 3 with 1 coming in. So that will help you tell what order they are going to go in. So based on that information, because this one in A is the longest, we know it's going to have to go last. And here we see that the blue one's getting kicked out, but the blue one's still attached here. That means that C has to come first. As blue, teal is coming in to join the party. We shift over. Blue gets kicked out through the exit site, and the teal is staying in the middle. Green comes in on A, getting ready to go to the next step. So we have CBA, number 3. Yes, thank you. Awesome. So these questions, the reason it says CQ3 is because they're supposed to be clicker questions. In the classroom, we don't use the clicker remotes, but we have little, either you use the dry erase boards or we'd have flashcards, and everybody would be holding up their answers for this. So, but that means that if other people aren't chiming in with their answers, if you don't tell me that you're having trouble, I'm going to assume that you're okay. So please feel free to communicate if you have any questions. All right, so 13, deoxytetanolide, I think that's how we say it, is an antibiotic that binds to the E site of the ribosome. If 13-deoxy-T is added right before translation starts, which one of the statements is true? Would translation not happen? Would translation would not be affected? The end product carries the antibiotic before the first amino acid methionine? Would the end product only have 2 amino acids, or would the end product only have 1 amino acid? So remember, E is the exit site. So how far would we get before it's going to fall apart because it can't ratchet forward? So I'm going to tell you, it's not going to be that translation would not be affected. It would not be a very good antibiotic if it didn't affect anything. It's not going to be able to just carry it along with it. The correct answer is going to be D, that the end product only has 2 amino acids, because you would get the first AUG start site lines up at the P site, not at the A site, lines up at P. You get methionine put into place, and the E site can sit there with the antibiotic. The next amino acid comes into the A site, but we can't ratchet forward after that. So we can't, because we can't release the E. So you would get the AUG in the P site, whatever the next amino acid is, and it couldn't go on any past that. So you get 2 amino acids. Now we're going to actually talk about FOXP2 protein, since we've done our review here. Now again, FOXP2, not italicized because it's not a protein, is found in certain but not all brain cells in the same individual. How is it possible? Is A, some brain cells don't have chromosome 7. B, FOXP2 DNA is only present in some brain cells. C, some brain cells don't have ribosomes. D, FOXP2 mRNA is only produced in some brain cells. Or E, some brain cells contain more DNA. Okay, so this is a question to remind you that all the cells in your body have the same DNA. So you better not have any chromosome or some brain cells without chromosome 7. You'd be dead, point blank. You better not be only having some of your DNA in some brain cells. Again, all of your brain cells, all of your body cells contain the same DNA. All of your cells better have ribosomes, because you need to have ribosomes to make proteins. Some of your brain cells should not contain more DNA, should have the same DNA in everyone. So that just leaves D, that the mRNA is only produced in some brain cells. So we get this because there's transcriptional regulation. As your body is being formed, you have a single cell that splits into 2, and then based on what it's exposed to, usually different hormone signals, it'll decide what kind of cell it's going to turn into. So in this case, you have it, some went to skin cells, some went to muscle cells. The other side of this is that you get temporary changes that are, in this case, like it talks about a stress response. So when there's no stress going on, it's not going to make the stress response proteins. When you hit a period of stress, it's going to turn on those genes to start making stress response. So like in bacteria, a lot of times they talk about heat shock proteins and things like that help it survive during periods of high heat. If it doesn't have high heat, you don't want to waste your resources to make those. So now as the brain is developing, normally the areas of the brain that should be making FOXP2 would be like in the Broca's region, but it's obviously not working right in these individuals. Now we are coming to part 2, so I'm going to pause the video here.